Hello everyone, my name is Ken, and today we're going to be talking about our skeletal muscle physiology lab, which is laboratory number 5. You could find the details about this in our lab manual from pages 47 to 61. The first type of muscle activity within a skeletal fiber that I want to talk about is called a twitch. And twitches are defined to be a single muscle contraction induced by a single action potential. Okay? In the figure to the right, you have in black what's supposed to be action potentials or stimuli, and above them are muscle tension. So we have a single action potential and the resulting contractile event. This is a twitch. On screen, what you should know is that we see two twitches based on that definition. To take a closer look into a twitch, what you'd know is you could divide a twitch into contractile event and relaxation event, where the tension in the muscle returns back to its baseline. And you might also notice that a single action potential starting at time point 0 does not automatically induce the contractile event, that there's actually a small delay that we call the latency period. In muscle physiology, when an action potential happens, there is a small period of time where calcium needs to be released within the cell so that they can bathe the sarcomeres within it, and then we begin excitation-contraction coupling and contractile events. That window in which calcium needs to be released is our latency period. When two twitches are happening because of the result of two action potentials seen here, and they are within a close enough proximity of time, this first twitch will not be able to get back to its baseline as you've seen in the previous slides. Instead, because of a second stimulus event right here, we end up inducing the muscle to contract and tension to increase even further. Okay? This event, in which two twitches are within close enough proximity and they build on each other, is an aspect of summation. Okay? And we're going to see two types of summation that can result. If twitches are like a Lego block, then this is us trying to stack them. Here is the big picture. Let's look at the bottom left where we see something a little more familiar, a twitch. A single stimulus, which in this case S represents an action potential going off, and then the resulting contractile event. The only difference between a twitch and the ability to start summation, which is seen in the later part of this figure, is we change the frequency in which stimuli are happening within that muscle fiber. So, if we took a twitch and started trying to stack twitches by increasing frequency, the first event that you'd see physiologically and observe is actually called unfused tetanus. So let's take down that word and try to figure out what it means. What you have are twitches that are building up on each other so that they, right when they're trying to get back to baseline, another action potential happens and further tension increases. Okay? We actually also call this TREP. There are actually three ways that we can refer to this type of phenomenon: unfused tetanus, TREP, and incomplete summation. You could think of it as incomplete because you can actually still see the individual twitches within this event. Another way to recognize TREP, or unfused tetanus, is there is something that we call a rising baseline. If we look right where this twitch ends and continue to sort of trace our finger down this line, we see that the following twitches are higher, and they're increasing in height. Another way to know that this is TREP is that TREP is German for steps or staircase. And typically, when you're seeing this and observing this, it does look similar to taking steps up a staircase. That rising phenomenon is extremely critical to know that you are seeing this unfused tetanus. Okay, twitch, and then this area, which is called TREP or unfused tetanus or incomplete summation. And then finally, to get to the final step, we must increase the stimulation of the muscle cell and cause fused tetanus, also known as complete summation or tetany. Fused tetanus refers to the fact that the twitches are no longer independently visible. They have all fused into a straight line. Instead of a staircase, it's more like the incline of a mountain, and it all ends up plateauing. Twitches right over here, as the result of a single stimulus, are actually quite uncommon. We don't tend to portray twitches. You might iconically think about eye twitching or irritation, but really they are uncommon. When you are moving your skeletal muscle, when you're getting up out of bed and moving and exercising, it is all very often in the form of fused tetanus. Another way to view this entire building of this type of physiology is you could see a twitch as a single monomer, a single unit, and if you place twitches close enough, you can get to summation. Either it will be incomplete summation, in that you still see each twitch independently, or fused tetanus, also known as complete summation. Now to change gears a little bit. If I were to pick up a pen and then place that pen down and pick up a 50-pound weight, you actually may do this as well, and you might feel that there is an engaging of more musculature within your arm. Okay? We actually divide muscle units into what we call motor units, which is defined to be a single alpha motor neuron and all the muscle fibers that motor neuron innervates. With that definition in mind, what I'd like you to do is try to guess how many motor units you see on screen. And if you actually guessed 2, you'd be correct. We have 2 alpha motor neurons in blue and orange, and then we have 2 sets of muscle fibers that are innervated by both sets of motor neurons. Now, the final part of this presentation is actually a technical guide to how you should be dissecting your frog's leg. Because we are looking at skeletal muscle, we are going to be handing each of your teams a single frog leg, and you're going to need to dissect and prepare it for use. The video will just explain exactly what you should be doing, how you should be doing it, and how you should complete your setup. If you have any other questions, feel free to comment or just ask me in person if you're there. Okay, thank you for listening.
Okay. All right. So the muscle that I just tore to pieces is actually something that we won't be needing. It is the quadricep, and it's something that if you wanted to, after this step, you could cut away. Just do that right now. Because what you're going to look for, what you need, is the muscle right here. Okay? As the gastric, what now? Nemius. The gastric nemius. And take a glass rod and separate the gastric nemius from the tibia fibula, like so. That way you can actually thread a thread right between the two of that, right between the bone and the muscle itself. And we'll show you that part right now. Okay. When you take your thread, it is advisable that you make sure it's a double thread. Fold it over, because you want something that's a little bit stronger for what we're going to be doing. So, and then you're going to thread the thread through like this. Not too tight that you actually cut through anything with the string, but it doesn't hurt to double knot it. After you have tied that, grab some scissors. You can take the glass rod out and feel free to place the scissors like so and cut away, so that you have your muscle, you have it attached to a piece of string, and then attached to the bone. You're not going to be needing the rest of this part right here, so let's go ahead and cut that. If the scissors are broken, then you need to get some new scissors. I am professional. So this is what your end result should look like. Maybe it's a little cleaner than mine. Congratulations if that's the case. But you have your femur right here, which I'm holding on to. You have the gastrocnemius attached to a piece of thread. We're going to place the femur right here in your little unit, your little housing. It's called a femur clamp for the reason that we're doing right now. And you just want it to be held there. Pretty legit. It's not going to go anywhere. This is your setup. The thread, the other end of it, is going to go through this doodad, which has a fancy word that I can't remember at the moment. So let's put this like so. You want to make sure that when you tie this other end of the string that there is some amount of tension on the whole overall attachment from the muscle to this unit down here. That means you might want to pull a little bit so that this rod comes up just slightly, and then feel free to tie it like so. Okay. You can also move the overall unit up and down. You don't have to like tie the string in this way, because if you want it to be more taut, you could just move the units holding the clamps. But basically, after you're done, you'll have a setup that looks something like this. The final part is making sure that the electrodes, which you can see right here, are actually placed in the muscle itself. You have your red and your black. The way that they're placed in, or which one's above the other, is not important as much as the fact that they're both sort of set into the muscle so that there can be a circuit passed through it. And let's bring that closer, reducing a little tension. So this is what your overall setup should look like, give or take. And yeah, after now we go into iWorks.